Hello everyone, welcome to the channel Microbial World. So today we will be discussing about bright field microscope. So the bright field microscope, it is also called as compound microscope. And these microscopes, they use light rays to produce a dark image against bright background. Okay, so this is the main reason why it is called a bright field microscope. The background, it will appear white in color, or the background is very bright, whereas the image of the specimen, it will be dark. And this is a standard microscope. This microscope, it is used in biology, cellular biology as well as microbiology laboratories. This microscope is used, and it is used to view fixed and live specimens. So now let's see the principle of a bright field microscope. So you can see here there is a source of light. The light first passes to the condenser lens. The condenser lens will focus the light onto the specimen, and then the light that is coming out of the specimen will be magnified by the objective lens, which is further magnified by the ocular lens or the eyepiece, and then the observer, it can view the image. Okay, so a uniform amount of light, a uniform beam of light is passing through this specimen, and the specimen, it will be stained. Why we are staining the specimen is because by staining the specimen, all the structures and components within that specimen will be clearly visible when we stain the specimen. Okay, and also it will increase the contrast of the image. It will increase the contrast of the image. What is contrast? Contrast means we can differentiate that specimen from the surroundings. We can highlight and see that specimen. So by staining, we can increase the contrast of the image. Now, when light passes through the specimen, what happens is that the light, it will interact with the structures and components within the specimen. So some parts of the specimen, that is the denser areas of the specimen, it will absorb some amount of light. It will reflect or refract the light, whereas some other parts, it will just allow that light to pass through. So the image that we get will be a dark image against a bright background. Now this is how we will get the image. Okay, so you can see here the background, it is very bright. The background is very bright. Okay, whereas the specimen, it is appearing dark. The specimen appears dark in color. Okay, this is how the image will be formed. And another thing is that the specimen, it is viewed under oil immersion. Okay, that is, we will place a drop of oil, cover it with a cover slip, and then view it under the microscope. So this is the principle of a bright field microscope. So now let's look into the parts of the bright field microscope. So you can see there at the bottom of the microscope, there is the light source producing light. Okay. Then there is the arm of the microscope. So this arm, it is like the backbone of the microscope. We just have to hold on to the arm so that we can carry and move this microscope from one place to another. And also at the bottom, you can see the base of microscope, which is like a stand for the microscope. We can also hold on to the base in order to move the microscope. Okay. Then below the stage, there is the condenser lens. So the condenser lens, as we have said earlier, it will focus the light onto the specimen. And just below the condenser lens is the diaphragm. The diaphragm's function, it is to control the amount of light or diameter of light that will pass through the condenser. And below the objective lens, you can see the stage. So the stage is where we will place the specimen. And the revolving nose piece, it is having 2 to 4 objective lens. So we just have to rotate this nose piece in order to select the objective lens with desired magnification. And what the objective lens does, it will magnify the light that passes through the specimen. Okay. And on the side, you can see there are 2 focusing knobs. There is coarse focus and fine focus. So this coarse adjustment knob, it will help us to move the stage up and down quickly. And the fine focus, or the fine adjustment knob, it is used for precise focusing, that is to increase the sharpness of the image or the clarity of the image. And on top of the microscope is the eyepiece or the ocular lens. This is used to further magnify the image that is produced by the objective lens so that we can view the image comfortably. So these are the main parts of the bright field microscope. Magnification of a bright field microscope. So the magnification of an image, it is determined by the magnification of objective lens against the magnification of eyepiece. So this objective lens, it is having a magnification power of, it ranges from 40x to 1000x depending upon the type of bright field microscope. Whereas the magnification power of eyepiece, the range is 8x to 12x, but it is having a standard magnification power of 10x. So the total magnification power is equal to magnification of objective lens into magnification of eyepiece. Now, if the magnification power of the objective lens is 45x and if the eyepiece is having, eyepiece we said it is having a standard magnification power of 10x. So 45x into 10x, 450x is the total magnification power. The advantages and disadvantages of a bright field microscope. The advantage is that it is very simple to use and operate. Therefore, it is used in the educational and research settings. And it is a widely available microscope, so it is a very good choice for the scientists and students. And these microscopes, they are less expensive when compared to other microscopes like electron microscope, confocal microscope, and it is very easy to modify. We only need to make a few adjustments in order to view the image. And it is good for observing the details. We can observe the fine details of the specimen. The disadvantage is that it is having limited contrast. So as a result, we have to stain the specimen in order to increase the contrast of the image. And also it is having low resolution when compared to microscopes like phase contrast microscope and electron microscope. And the use of oil immersion might distort the image. Using cover slip can damage the specimen. And also these microscopes, they are using strong light source. So these light, it will generate a lot of heat and it might damage or kill the specimen. And we are also staining the specimen to increase the contrast of the image, but this staining, it might introduce certain unwanted materials into the specimen and contaminate the specimen. So now let's see some of the applications of bright field microscope. So bright field microscope, it is used in biology and medicine. It is used to study the structure and function of cells, tissues, organisms. These microscopes, it's used in blood counting, to study about the animal cells, plant cells, examine the bacterial cells, fungal cells. Okay. Then it is used in pharmaceutical industry to analyze the purity and quality of the drugs. Okay. They will find the contaminants and impurities that is present in the drugs so that those drugs can meet quality standards. And in the food industry, these microscopes, they will detect any contaminants or defects that is present in the food products. In the field of geology, it is used in the identification of minerals and also to study the microstructure of rocks. In forensic science, it is used to analyze the evidence for criminal investigations, to identify hair, small particles which can be used as evidence in the investigation. And in education, these microscopes will introduce the principles of microscopy to the students, so that the students using these microscopes, they can understand the structure and function of cells and tissues. So these are some of the applications of bright field microscope. So this is all about bright field microscope. I hope that this topic was clear for everyone. Thank you.